Hello students. So Mendelian experiment on dihybrid cross. What is dihybrid cross and what about the effect what about the results of dihybrid cross, that I will explain now. Dihybrid cross means I have taken two pairs of character, means the Mendel has taken two pairs of characters. See, capital Y, capital Y, capital R means pure yellow round. Seeds, we have taken seeds. So these are pure yellow round. So one character that is yellow, other one is round. Similarly, here pure green wrinkled seeds we have taken. So green and wrinkles. So how many characters? Two pairs of characters here present. That's why it is known as Mendel experiment of dihybrid cross. Now dihybrid cross very important. Why? Because in dihybrid cross so many varieties are coming out. See, here it is capital Y, capital Y, capital R, and we are getting from here capital Y, capital R, because 2Y, 1Y will come, and from 2R, 1R. So that is yellow round, and from here we are getting the character small y, small r, that is green wrinkled seeds. And in F1 generation we are getting the character capital Y, capital R, small r. So phenotype, according to the phenotype character, the seed that is capital Y, capital R, and similarly small y, small r are also present. Here capital Y, capital R, these are dominating character, and small y, small r, these are recessive character. So capital Y, capital R, it will suppress the characteristic features of small y, as they are recessive character. So here we are getting the seeds that sees capital Y, capital R, as they are dominating the character of small y, small r. So this is known as yellow round seeds. So in F1 generation we are getting the character of capital Y, capital R, small r, and there will be cross. And after cross there will be four combination. Four combination. How four combination will comes out? See the structure, that is capital Y, small y. So there will be cross. So you can write it from capital Y, capital R, capital Y, small r, capital Y, capital R, capital Y, small r. Because starting from here, skip that Y, then write capital Y, capital R, and then capital Y, small r. Similarly, here you will start from small y, so small y, small r, small y, small r. So look at the structure. Here I am starting from capital Y, small r, capital Y, small r. So how can you write it? If you want to write it, so take that y and skip that small y, then you can write capital Y, capital R, capital Y, small r. So capital Y, capital R, capital Y, small r. Similarly, starting from small y, capital R, small y, small r. So it's a very easy tricks for you. So these are combination of dihybrid cross. Now to explain the dihybrid cross, I will show you the checkerboard. If you cross that dihybrid cross, means capital Y, capital R, small r, along with that F1 generation capital Y, small r, capital R, small r. So you will get that possible combination. What are the possible combination we are getting? These are the possible combination. And if you think these are male gametes, similarly if you think these are male gametes, I have written here. So same female gametes here present here. So these are male gametes, these are female gametes. So we are getting the characteristic features. How? Because capital Y, capital R. So here capital Y, capital R. So capital Y, capital Y, capital R, capital R. Similarly here capital Y, capital R, small r. Here capital Y, small y, capital R, capital R. So you can write the combination. We are getting 16 combinations are there. Among 16 combinations, look at first combination, that is capital R, capital Y, capital Y, capital R, capital R. So the structure which is present here, that is capital Y, capital R, capital R. The structure that is yellow round seeds. Similarly, it is also yellow round seeds. It is also yellow round. It is also yellow round. Why? Because capital Y, capital R are present, and these are suppressing character. These are dominating character. These are, so it is yellow round seeds. Here also yellow round. But look at this combination. It is capital Y, smaller, smaller. Means capital Y means that is yellow. The seed is yellow. But it is small r. As it is small r, so it is wrinkled. So yellow wrinkled. Similarly, it is also yellow wrinkled because due to capital Y, it is yellow, and due to as no capital R's are present, so it is not the round character, it is wrinkled character. Similarly, here is also yellow wrinkled seeds is also present here, yellow wrinkled. Again, see the structure, that is small y means not the yellow character. So if the yellow character is not here, there is also green character, because with the help of small y we represent green character. So here the structure, that is green character and the round seeds. So that is green round seeds. Similarly, that is also green round seeds. So the combination we are getting yellow round seeds. These are 9 combination. 1 2 3 4 5 6 7 8 9 yellow round seeds. How many combination? 9 combination. And the structure which we are getting here, that is yellow wrinkled seeds. How many yellow wrinkled? That is 3. Yeah, we are getting the structure 1 2 3, and green round seeds 1 2 and 3 green round. So these are also another combination. And the last one, just only one character, that is green wrinkled character. That is a recessive character. It is not suppressed, it is ultimately suppressed character, and we are getting that character. So if you see the phenotypic character, the phenotypic character is 9:3:3:1. Why? Yellow round seeds is to yellow wrinkled seeds is to green round seeds is to green wrinkled seeds. That is 9:3:3:1. And the genotypic character, it is very complex, so I will not explain it now. In genotypic character, it is not in class 10 syllabus. So just up to phenotypic character of dihybrid cross, that is 9:3:3:1. It is about genotypic character regarding phenotypic character regarding dihybrid cross. So after Mendelian experiment, Mendel has given 3 laws. There are law of segregation, law of independent assortment, and also law of dominance. What is law of dominance? See the structure here. The structure means the yellow round seeds. It is suppressing the characteristic features of pure green wrinkle seeds. So the out of 2 pairs of characteristic features, 1 pair is suppressing the characteristic features of other. So that is law of dominance. What is law of segregation? Law of segregation means the pair of factors of genes contrasting or identical in nature present in an individual of any specific character is segregated during gamete formation, and each factor goes into one gamete. So law of segregation means when the gametes are coming, they are also segregating. They are capital Y, but 2 Y separated, only 1 Y is coming here. So they are also segregating. That is law of segregation. And what is law of independent assortment? That is 2 or more pairs of contrasting factors or genes for 2 or more characters of an organism segregate independently during gamete formation, appearing in all possible combination in the gametes. Means here from capital Y, capital R, only 1 Y is coming here. So that capital Y is representing the whole entire characteristic features of that Y. So here it is represent at it is representing the characteristic features of the whole of that yellow seeds. So that is law of independent assortment. We are getting the character from Mendelian dihybrid cross. It's a very important. So three laws from Mendel we are getting. Now these are Mendelian experiment. Students, I will show you the opposite or incomplete codominance. Ultimately, incomplete codominance means which is not ultimately maintaining the characteristic features of Mendelian experiment. To explain this portion, we have taken Mirabilis plant, and from Mirabilis plant how incomplete codominance will occur, and from incomplete codominance how the Mendelian experiment will not follow here, that I will explain. Here just we are discussing about Mendelian experiment, and these are some Mendelian experiment, and now I will discuss about the opposite, means the experiment which will not follow the Mendelian experiment, that is in some Mirabilis plant. In Mirabilis plant, if these are red flower and these are white flower, so it will come capital R, and from here capital W means white flower. So when we will get the structure of capital R, capital W, so either capital R is dominating character or capital W will be dominating character. So in that character, one will be dominating, other one is suppressing. Either the flower will be red or either the flower will be white. But here the flower will be mixture color of red and white, or purple you can say. Why? Because the characteristic features, that is red color, and the characteristic features, that is white color, they are ultimately mixing with each other. This kind of experiment known as incomplete codominance, where it will not follow the Mendelian experiment. And from here, that is capital R, capital W cross capital R, capital W. So there will be the product. If you do Mendelian, if you do the checkerboard, so there will be capital R, capital R, capital R, capital W. So similarly, capital R, capital W, W, W. So look at the structure here: capital R, red flower, capital R, capital W. These are which kind of flower? These are purple, and these are white flower. So phenotypic character, genotypic character same, means it is 25% red color, it is also 25, it is also 25, means purple flower 50%, red flower 25%, and white flower also 25%, it is 25%. That's why the phenotypic ratio here 1:2:1. Also the genotypic ratio will be 1:2:1. These are incomplete codominance. Last topic, that is X-linked inheritance. What is X-linked inheritance and what are the experiment regarding to X-linked inheritance? X-linked inheritance means sometimes we have listened about some diseases just like hemophilia, color blindness, and if our parent affected by color blindness or hemophilia, so what will be the effect, that I am explaining now. Look at the structure. These are known as X-linked inheritance. It is known as X-linked inheritance. Why X-linked? See, XX, these are female, and XY, these are male. So among this pair, if the female one X chromosome is affected by hemophilia, means it is hemophilic. So if there will be cross, so what will be their products? So ultimately after their cross, the product will be XXH XX, yeah, XHY XY. Look at the structure, the products will be looks like that. Means here XX, one X is affected. In female, two X chromosomes are there. If one is affected and the other one is normal, then the female is carrier. She will not affect it, but she is not hemophilic, she is a carrier. And if the father is normal, so if there will be sexual intercourse, after that the possible combination between their offsprings, these are like that. See here, I am starting from that structure. Here is the structure, that is XH, is the structure, that is XH. One X is affected, other one is normal, means the daughter, that is carrier, not fully affected. If you see any person that is carrier of hemophilic, so you will not get any